First, you should be able to list the structures and those functions of those structures in the human brain. You should be able to describe how electrical and chemical information is sent in the nervous system. You should also be able to talk about the mechanisms that are involved in altered states of consciousness. So how is it that we see changes in memory because of hypnosis or meditation? How does sleep and dreaming work? And how do drugs of abuse alter consciousness, for example? Lastly, you should be able to analyze your personal sleep habits and come to conclusions about what you might do to improve those sleep habits. To get us started though, let's actually talk about the brain and what we know about it. One of the things that's fascinating is that our current understanding of the brain is very different than the understanding from just even a few hundred years ago. For example, ancient Egyptians believed that the brain was really a radiator of heat, and that in fact it really wasn't that important. You see evidence of this in that the brain was discarded before they would mummify pharaohs. And in fact, what they would keep, right, were the heart, the kidneys, other major organs. They in fact believed that the soul was actually kept in the heart. For them, what early physicians noticed was that a small nick to the heart would actually kill a person, but yet large areas of damage to the brain really didn't produce substantial loss of function. In fact, current evidence shows that you can lose about half of your brain, or at least in cats, we can remove about half of their brain, and they're still able to function. You really wouldn't notice a difference. Now, this may say something about cats, but that's because I'm a dog lover. Anyways, fast forward a few hundred years, and what we see is that Galen of Pergamon, who is often credited as being one of the first physicians, he proposed a much more complicated idea about the brain. At this point, he clearly recognized that the brain was important in basic cognitive function and sensory functions. He suggested, in fact, and you can see this in the picture on the side there, that information came in through the auditus, gustus, olfactus, tactus, and visus systems. Those are your sensory systems. And he believed they were processed in the front of your brain in an area he called the sensus communis. So all this sensory information comes together. Then it's passed in steps to more complicated areas of the brain where fantasy, imagination, and finally cognition, estimation, and memories. He believed that memories were stored in the very back of the brain. Now, the locations of those functions are not correct, but he is on the right path trying to understand that the brain is really important in a lot of very complicated functions that we need it for on a day-to-day basis. Then what we see is a big change. So during the Dark Ages, the Catholic Church put very strong restrictions about dissections of human bodies. And so we lost a lot of that early information that Galen and others had suggested. And then it wasn't until the Renaissance, around the 1600s, that we started to see people doing dissections again and starting to really think about what the brain did. Fast forward to 1809, and Franz Gall suggests that in fact each part of the brain is an important contribution to a different piece of your personality, okay? So for Franz Gall, each different part of your brain told you whether you would be kind or friendly or greedy or confident. That idea has been discredited. What he originally proposed was that bumps on your skull were related to the underlying brain matter. Bumps on your skull really have more to do with whether or not your parents dropped you on your head than they have anything to do with the personality that you may have or the size of the brain underneath, whether it's large or small, okay? So let's keep going though, but Franz Gall's idea that you can localize certain functions to specific parts of the brain is really important. So I have a small game we're going to play now. These are 6 different brains and 6 species of animal. I want you to see if you can figure out which brain goes with which animal. Some of them are going to be easier. Some of them are much more complicated. I'll give you some time to do that. Okay, were you able to figure it out? So of course, I'm sure you know that the lower left-hand corner is the human brain. This one though, look at this one. Look at how flat it is. So that's actually the brain of a possum, marsupial. You can see that it doesn't have the same kind of wrinkles that the other brains do. This is because actually their brain is not as complicated in some ways. You can see they have a very large olfactory bulb, which is used for smelling. This is the brain stem. You can see they've got a pretty large brain stem back here. And you can compare, you can see the brain stems in these other ones, right? This right here is the dog. Right here next to it is a zebra. You can see that very large brain stem and the very large olfactory bulb here in the zebra and in the dog. Then this is a dolphin, and you can sort of tell by the shape of it, right? Your skull shape really often determines what shape your brain is going to be. You can see the brain stem down in the back here. You can't really see the olfactory bulb, nor can you see the olfactory bulb that's very tiny here in the human. And then this is a rhesus monkey. You can see, like an opossum, it's very smooth, it's not very wrinkled. The way you can think about it, so this outer surface of the brain, the wrinkled portion in most of the brains that you're looking at, is the one that has developed most recently evolutionarily. Okay, so it's the one that is the most constrained in how big it can get. Right, early on, evolutionarily, your skull, like you can see in the opossum, there was a lot of room, so the brain didn't become wrinkled. The brain essentially starts in the middle, and then evolutionarily over time we added additional layers on the outside. So as species started to grow larger brains, the problem is their skull is limited in how big it can be, and that limits how big their brain can grow. So as you try to add more brain matter on the outer edge of a brain, like in a dolphin or a zebra or a human, it's essentially the same thing that happens when my son tries to put his clothes away, is that he just basically stuffs everything in to try to get as many clothes in one drawer as possible, and he just ends up getting a bunch of wrinkled clothes. So you get wrinkles in the human brain and other brains because you're trying to add additional layers on the outside of the cerebral cortex, which is the outer surface of the brain, that gives it that wrinkled appearance. Let's take a closer look here at the human brain specifically. So this is a cross section, so we've cut the human brain in half. So the eyes would be right here looking in this direction. This is the cerebral cortex on the outer edge here on the top, and you can see how wrinkled it is, right? Whereas deeper down you can see it's not as wrinkled, right? This is the hindbrain back here. The midbrain is this small structure right here, and then most of the human brain is actually devoted to the forebrain. In general, we actually divide up all brains in all mammals, reptiles, and other species into hindbrain, midbrain, and forebrain. The hindbrain, it does a lot of your basic life functions, so it regulates sleep cycles, it regulates alertness, it regulates breathing, heart rate, stress hormones. Basic things you need to survive are all regulated through the hindbrain, basic motor function, stuff like that. The midbrain does some regulation of more complicated things, such as movement and coordination of movement and sensory systems. So whenever you hear your name and you turn your head to see who was talking about you, the coordination of moving your head after it's activated your hearing, that is something that's responsible for, that comes from the midbrain. Then all the more complicated things, especially in humans, are happening in the forebrain. And you can see in humans the forebrain is the largest. The middle structures here are part of something called the limbic system. So the limbic system regulates emotion, memory, emotional recognition, fear processing, some hormonal processes. So ovulatory cycles and sexual mating cycles are all regulated in some of these areas here. Then on the very top part, right, the cerebral cortex, that does probably the most complicated functions. And let's take a look at a different image to look at that. So now, again, we're looking at the outside, the cerebral cortex, it means outer bark of the human brain, and it's generally divided up into 4 lobes. The frontal lobe, which is in blue, the parietal lobe, which is in green, the occipital lobe in red, and the temporal lobe, which is in orangey brown, burnt sienna. I'll let you figure that out. Anyways, so the frontal lobe is the largest in humans. It does a lot of planning, personality, motor coordination, language production, right? All of your complex motor functions, the ability to skip, the ability to drive a car, to know how to type, all those things initially start out, you learn those with the motor cortex. Your prefrontal cortex, which is another part of the frontal lobe, regulates impulse control and attention, right? What allows you to keep your attention up right now in like minute 8 of this video. I know sometimes these lectures kind of drag on, but right, it's really important for your brain to be able to tell you, oh, wait, this is important. I need to keep paying attention, right? So that's something that's regulating your prefrontal cortex. There is some evidence actually that people with ADHD have problems with impulse control in the prefrontal cortex, right? So that would make a certain amount of sense. Right behind the frontal lobe is the parietal lobe, which regulates your sense of where your body is located in space, your sense of touch. If you've ever heard of phantom limb syndrome, where people have sensations in a limb that they no longer have. So they're an amputee, and yet they still have sensations like someone is touching their hand or holding their hand, or maybe they have burning sensations in that hand that they no longer have. That's because of something going on in the somatosensory cortex. Okay? In the back is your occipital lobe. So your occipital lobe regulates vision, right? So it's the one in the very back, which Galen suggests regulates memory, but in fact, evidence tells us that it's really important in visual sensation, recognizing lines, vertical lines, horizontal lines, to recognizing faces, all of that, from the most simple kinds of objects to the most complex kinds of objects, all comes from the occipital lobe. And then lastly is the temporal lobe. So the temporal lobe has your auditory cortex, so it's recognizing language, even if it's sign language. Actually, you're using your temporal lobe to do that. If you're understanding what people are signing to you rather than just gibberish signs, you're using your auditory cortex to do that. If you have perfect pitch and you can recognize different musical notes, that's also something regulated from the temporal lobe. What's fascinating is that, in fact, we know a lot about these different brain areas, and some of it comes from case studies, these small studies involving an accident that happened to one person. For example, there is a man who had a small stroke that damaged his ability to see faces. And what's fascinating is he could see everything else in the world. He could see neck, hair, jewelry, but he could not see a face. His brain would essentially block it out and couldn't recognize what it was. Even if it was his wife, he could recognize her voice, but he could not see her face. If he were staring at her, what he would see is essentially a blank spot where a face should be. But he was able to figure out who it might be by using other cues, right? So he was able to adapt because he would recognize the voice of his wife, or what jewelry she was wearing, or the shoes she had on, and he would use those cues to be able to figure out who might be talking to him. But while that's important in recognizing close friends, you can imagine how on a day-to-day basis, going to work, for example, like if you work in customer service, you probably wouldn't even be able to function really well because you wouldn't be able to maintain eye contact. All these other things that we take for granted, you wouldn't be able to do necessarily, right? There are a lot of cases like this where people lose very tiny pieces of functioning because of small areas of damage to the brain. So Franz Gall, in some way, right? Remember that phrenology where the bumps on your skull predict your personality? He was kind of right in that tiny parts of your brain really do regulate tiny functions, right? Now, we've been talking about sort of macro anatomy, the big parts of your brain, but in fact, all of your brain is actually made up of tiny neurons and other kinds of brain cells. This is one example of a neuron. There are many different kinds of neurons. They don't all look like this, but in general, they're composed of a certain set of pieces. Neurons are specialized to move information. This one in particular moves information long distances. So information comes into the dendrites, these branch-like structures at the end of the neuron. It then converts, for example, light information in your eye into an electrical signal. That electrical signal is then sent down through this long tube called an axon. So it's able to get information about lighting in a room, for example, all the way down into your brain so that then your brain can start making sense of what you're seeing, okay? So it's kind of a fascinating, just a very basic change in electrical physiology causes you to then be able to recognize faces or shapes or your car, or whether it's your mom or your father. All these things are regulated by a pretty simple process, okay? In the middle of this cell is, of course, the cell body. If you remember your biology from high school, it has all the normal kinds of cellular machinery, including DNA. It's got ribosomes, endoplasmic reticulum, all those other things you may be studying right now in your biology class, for example. Okay. Your axon, again, as I mentioned, is a long tube that's specialized to send information long distances. It's made to get information quickly to places, okay? So if I go into my daughter's room and I step on her Legos and I'm cursing and screaming, one of the things that's fascinating is that that's sending an electrical signal up through my foot to the bottom of my spinal cord. That then tells me, ow, you need to move, which then triggers a muscle response so that I will move out of the way, okay? That happens in less than a second. It is a very rapid response. But actually, that rapid response would not be possible without a white substance that wraps around all of your axons, or most of your axons, called myelin. So your axon is sort of like a leaky hose, that as water is going down the hose, it's pouring out the sides. So if you were actually trying to water your plants with that leaky hose, you wouldn't have a lot of water pressure at the very end of that hose because you'd be losing most of it along the way. In fact, actually, myelin is sort of like duct tape. In sort of a simplistic example, myelin is like duct tape that's wrapped around that leaky hose, so it allows water to flow more consistently, or in this case, the electrical signal to flow more consistently over these short distances, which then gets it all the way to the end, to these terminal buttons, okay? Those terminal buttons then send an electrical signal, or a chemical signal, sorry. That chemical signal then is what triggers emotions, memories, right? All these sort of complicated changes in psychological functioning happen because we then convert that electrical signal into a chemical signal. So it might release chemicals like serotonin, dopamine, epinephrine, or what you think of as adrenaline, right? All those happen because neuron is releasing those. And neurons are really specialized to only release 1, 2, maybe 3 different kinds of neurotransmitters. So they're not capable of releasing all of them. And it just depends on the neuron you're talking about and where it's located in the brain. 20 years ago, if you had come and asked me, oh, what happens though as you get older and you start losing neurons, do you ever grow those back? One of the things that I would have told you 20 years ago, and what I learned in college, was that you can't regrow neurons. Once they're gone, if they're in your brain, they're gone forever. There's no getting them back. But in fact, actually, more and more evidence is telling us that in fact people can recover neurons in their brain. Not surprisingly, we have found evidence of stem cells actually inside the brain. And these stem cells are not just in babies, right? Babies, of course, it's not surprising they might have stem cells. They're still growing. They need those stem cells to make new brain cells. Fascinatingly enough, though, in adult humans, we see that areas of the brain that are needed to always be changing. For example, there's an area of your brain called the hippocampus. Hippocampus is located in this spot right here on the side of your brain. We found stem cells in the adult human hippocampus. The hippocampus is important for making new memories, so that makes sense. If you're going to make new memories, you probably need new cells to make those memories or to somehow solidify them. So it makes sense that you would want stem cells in these red areas. In addition, we've also found stem cells along the ventricles. These are fluid-filled area of your brain. The ventricles sort of produce cerebral spinal fluid that helps to keep your brain in a nutrient bath, and it also protects it from some injury. It's kind of floating there, so it's buoyed against car accidents or other kinds of trauma. Along the ventricles, though, you also have cells that have these long, almost like spoke-like protuberances, and those spokes actually are what your brain uses to guide stem cells to new locations. So in fact, along the ventricles, you're creating stem cells, and then your brain is sending them out along these spokes to wherever they might be needed in the brain for new memories or new functions. Any new skill you gain is probably causing you to develop new cells, okay? What's interesting, though, is this evidence originally did not come from humans. It came from canaries. So some songbird species actually will regrow entire areas of their brain. So they'll destroy it in the winter when they don't need that song area, right? Birds, for the most part, only sing in breeding season in the spring. So what's interesting is these birds in the winter, they destroy those song areas, and then when spring comes along and it's time to mate again, right, it's mostly males who are doing the singing, used to defend territories and attract mates. They will regrow, and you can see each of these black dots is a cell body. And so you can see that here in winter, these areas are smaller, where the arrows are. And then over here on the side, you can see that these areas are actually enlarged by the following spring, okay? That's not surprising, actually, though, because your brain is pretty energetically expensive. It takes a lot of calories to power a brain. And for birds who can't store any fat, because it means they can't fly, it's really important for them to conserve every single calorie. So that's why a lot of birds, for example, will store food over the winter so they can survive, because they don't have fat stores to survive off of like other species. In addition, though, these birds are also then saving energy by getting rid of brain areas that they only need at certain times of the year. This evidence is what led us to eventually look for stem cells in the human brain that now tell us that, in fact, you can regrow function. You can recover from a stroke, for example. So if you've had a stroke and you've damaged part of your brain, the more you practice with the muscles, for example, that might have been damaged in that brain area, the more you practice with those muscle groups that are connected to that brain area. So if you're paralyzed in your left hand and you keep using your left hand, you keep practicing with it, you actually can eventually regain function. Now, it's very slow. It may take decades to regain full function, but it is possible to regain function. Okay, so we've covered a lot of information in a very short period of time. I will leave you to take a look at the textbook. If you have any other questions about material or assignments, please make sure you send me an email. Thank you.